Hereby, I open this academic ceremony at Maastricht University. First of all, I'd like to welcome Mr. Nikolaos Taxiarchis Skenteris, and he will defend his academic thesis, Interplay between inflammation and calcification in cardiovascular diseases. And he will defend this thesis publicly as a part of a double degree of Maastricht University and Karolinska Institute in Sweden. And he will defend his this part in Sweden on coming Friday, June the 3rd. I would like to welcome all the members of the degree committee, in particular the 4 supervisors. And the supervisors are Professor Erik Biese, he is Professor of Experimental Vascular Pathology at Maastricht University. Professor Chris Reutlingsberger, he is Professor of Biochemistry of Apoptosis. And the 3rd supervisor is Assistant Professor Dr. Hildur Arnardóttir. She is Assistant Professor in Translational Cardiology at the Department of Medicine at the Karolinska University Hospital in Sweden. And the 4th supervisor is Associate Professor Dr. Ljubika Matic. She is also Associate Professor in Translational Cardiology at the Department of Medicine, Karolinska University Hospital in Sweden. And I will introduce the 6 opponents later during the ceremony. And I welcome, of course, all the followers of the live stream. Mr. Skenteris, may I invite you to give a summary of your research? Please go ahead.
Did you see my presentation? So, good afternoon, dear Prorector, dear members of the Corona, dear family, friends, and audience. In the next minutes, I will shortly summarize my doctoral thesis entitled as Interplay between inflammation and calcification in cardiovascular diseases. This is the outline of my today's talk. Cardiovascular calcification is an active pathophysiological process which is associated with atherosclerosis, diabetes, chronic kidney disease, and calcific aortic valve disease, as well as aging, and is linked with major adverse events. Calcification is classified according to the location as intimal and medial arterial calcification, as well as valvular calcification when the minerals are deposited in the aortic valve leaflets. According to size, calcification can be classified into 2 forms, named micro and macro calcification, which occur side by side during disease progression. Presence of microcalcification in the tissue is a sign of a dynamic inflammation triggering procedure. Highly inflamed plaques contain microcalcification as well as pro-inflammatory cells, which contribute to enlargement of lipids in necrotic core, as well as thinning of fibrous cap, rendering the plaque unstable and more prone to rupture. Several studies from our lab have shown that a presence of macrocalcification as well as osteochondrogenic smooth muscle differentiation are associated with more reparative healing processes and plaque stability. Calcification and inflammation are engaged in a vicious cycle in highly inflamed and stable plaques. However, in stable calcified plaques, inflammation is widely suppressed. Besides inflammation, the progression of calcification is a consequence of a complex mechanism, interplay between a broad range of determinants, such as lipid necrotic core, intraplaque hemorrhage, matrix composition and remodeling, and cellular heterogeneity. However, the underlying mechanism is still unclear, while no medical treatment is available to prevent or reverse the calcium deposition. Therefore, the current thesis aims to provide new insights into macrocalcification mechanism by means of molecular, cellular, morphological, and clinical data integration. More specifically, Study 1 aims to unveil novel molecular pathways and networks in calcification. Studies 2 and 3 aim to identify and functionally characterize markers of calcification and link those markers with future cardiovascular events. And Study 4 aims to understand the underlying mechanism between calcification and cellular phenotypic switching. To achieve this, we applied a complex integrative strategy that combines biobank of human specimens as well as medical records and diagnostic imaging, tissue gene expression profiling, as well as advanced computational omics and imaging analysis techniques, rodent models of vascular calcification, and cellular in vitro culture from functional mechanistic investigations. More importantly, through an international network of established collaborators, we had access to unique clinical data and patient material from large cohorts in order to study vascular calcification. Moving forward to the results, the first aim was to unveil molecular pathways and networks in calcification, which led us to study one entitled as Integrative Multi-Omics Analysis in Calcific Aortic Valve Disease reveals a link to the formation of amyloid-like deposits. Here, publicly available single-omic datasets from healthy and diseased individuals with calcific tricuspid aortic valves, further processed with bioinformatic analysis in order to identify a unique signature that explains the disease phenotype. Integration of proteomics and transcriptomic data revealed 9 common molecules, as you can appreciate in the panel, and integration of those 9 molecules with the 32 microRNAs from single omics revealed a highly connected 3D layered network, as you can appreciate from the image. The network contains 7 seed molecules as well as 4 miRNAs. In addition, it contains 14 molecules, connector molecules, the red nodes, which directly connect with at least 2 seed molecules. Those 2 molecules, APP, which is amyloid precursor protein, and TTR, transthyretin, connect directly with 3 seed molecules. And next, we assessed the presence of those 2 molecules in human calcified valves, and we found the presence of those 2 molecules in a calcified part of the valve leaflet. However, no expression was observed in the non-calcified regions. Interestingly, TTR aggregations observed in calcific aortic valves leaflets and displayed amyloid-like properties similar to those found in brain tissue of patients with amyloid angiopathy, cerebral amyloid angiopathy. To conclude, multi-omic data integration is a useful tool to identify a novel molecular network across layers and to correlate phenotype and network with the pathway network with the phenotype. In our study, we found a link between calcification process and amyloid-like deposits in calcific aortic valve disease. The second aim was to identify and functionally characterize markers of calcification and link those with future cardiovascular events, which led us to study 2, entitled as osteomodulin, attenuates smooth muscle osteogenic transition in vascular calcification, as well as to the overlapping study 3, entitled as osteomodulin gene expression is associated with plaque calcification, stability, and fewer cardiovascular events in the CPIP cohort. Osteomodulin is a proteoglycan which is involved in extracellular matrix mineralization both in bone and teeth. Recent studies have associated osteomodulin with cardiovascular risk traits and type 2 diabetes. Considering that osteomodulin is linked with mineralization and calcification is associated with diabetes, we hypothesized that osteomodulin could be a biomarker for vascular calcification. To achieve this, we followed a translational approach, integrating data from 3 human cohorts for carotid atherosclerosis, calcific aortic valve disease, and chronic kidney disease, as well as murine models for intimal and medial calcification and in vitro investigations. Here I will present only the data from carotid atherosclerosis and in vitro investigations. We found that osteomodulin mRNA expression was upregulated in plaques compared to normal arteries, and certification of microarray data according to the degree of calcification, as estimated by CT angiographies, we can appreciate that osteomodulin mRNA expression is upregulated in highly calcified plaques compared to low calcified plaques. Immunohistochemistry showed the absence of osteomodulin in normal arteries, while it confirms its presence intracellularly and extracellularly, both in low and highly calcified plaques. Particularly in high calcified plaques, osteomodulin is found surrounding macrocalcification regions abundant with the smooth muscle alpha actin. In addition, the osteomodulin expression is upregulated in asymptomatic patients compared to symptomatic ones, and further, following those patients for 120 months, we found that higher osteomodulin tissue levels are associated with fewer cardiovascular events and fewer cardiovascular death compared to those levels with low levels of osteomodulin. Next, we assessed the association between circulating osteomodulin from end-stage carotid atherosclerotic patients with plaque morphological characteristics and particular calcification, and we found that there is a positive correlation between plasma osteomodulin levels and calcification. To investigate further which cells express osteomodulin, we utilized publicly available single-cell RNA data of human carotid and coronary atherosclerotic plaques, and we found that osteomodulin is abundantly expressed for fibroblast and fibromyocyte-like cells, as well as for fibromyochondrocyte-like cells. Considering that smooth muscle cells are the main cell type that undergo phenotypic switching during calcification, we used coronary smooth muscle cells and we treated with beta-glycerophosphate. And we saw that osteomodulin mRNA levels upregulated throughout the course of experiment. In order to investigate further the role of osteomodulin in this experimental setup, we silenced the osteomodulin gene, and we found under beta-glycerophosphate conditions that extracellular matrix calcification was upregulating. At the same time, we used aortic smooth muscle cells and we treated with high phosphate conditions. And we found that osteomodulin mRNA levels upregulated quite early at day 3 during the course of experiments and reversed to basal levels or even downregulated at later time points. In order to assess further this investigation, we administer exogenous administration of full-length human recombinant osteomodulin, and we found that the matrix calcification was widely attenuated. Gene-set enrichment analysis revealed that pathways related to extracellular matrix, collagen and fibril formation, as well as smooth muscle contraction, were widely upregulated, where pathways related to osteoblastic smooth muscle osteoblastic transition were downregulated, such as Wnt signaling, BMP2, and Notch. And this is the conclusion of this study, showing that exogenous osteomodulin in osteogenic conditions maintains smooth muscle contractility and induces a protective matrix secretory reprogramming of smooth muscles, while it attenuates the osteoblastic transition and the matrix calcification. To conclude study 2 and 3, we found that osteomodulin as a novel marker related to carotid atherosclerosis calcification, osteomodulin mRNA levels are associated with fewer cardiovascular events, modulated smooth muscle cells express osteomodulin, and exogenous osteomodulin attenuates matrix calcification. The third aim was to understand the underlying mechanism between calcification and cellular phenotypic switching, which led us to study 4 entitled as mast cell participate in atherosclerotic plaque calcification and smooth muscle cell reprogramming. The rationale behind this study is that unstable lesions from patients with symptoms are enriched in activated mast cells, and in addition, mast cells are associated with plaque microvessel density in future cardiovascular events. Therefore, we employed an integrative system biology approach from the large biobank of Karolinska enterectomies BIKE in order to explore the link between mast cell activation and macrocalcification. Carotid plaques from patients were analyzed using global transcriptomic analysis, and the data further processed with CyberSoft software in order to estimate the immune cell composition. At the same time, CT angiographies from the same patients were further processed bioinformatically with VasoCap software in order to quantify the plaque morphological features such as interplaque hemorrhage, calcification, and lipid-rich necrotic core. The analysis revealed the abundance of activated mast cells in low calcified plaques, whereas high calcified plaques are enriched with resting mast cells. Multivariable analysis between activated mast cells and morphological features, as they were estimated by VasoCap software, revealed an independent negative association between activated mast cells and calcification. This association remained when we analyzed plaques only from symptomatic patients. Next, considering that activated mast cells are upregulated in enriched in low calcified plaques, we used a transfer methodology approach in order to further investigate this association. Facial isolated BIKE plaques, culture ex vivo for 24 hours, and the supernatants transferred on resting mast cells. And we assessed the activation of mast cells as a percentage of CD63 molecules. And we found that supernatants that come from highly calcified plaques attenuate mast cell activation, and at the same time attenuate cytokine release. However, plaques that come from highly lipid necrotic core plaque induce mast cell activation, and we test the same in smooth mast cells, and we saw that calcified smooth mast cells attenuate the mast cell activation, and we did the opposite. We transfer supernatants from either resting or activated mast cells on smooth mast cells in combination with high phosphate, and we saw that both resting and activated mast cells upregulate matrix calcification to a higher degree compared to phosphate only, and at the same time induce osteoblastic smooth mast cell reprogramming. To conclude this study, combination of transcriptomic and imaging analysis revealed the abundance of activated mast cells in low calcified plaques and resting mast cells in high calcified plaques. And this finding was further validated in vitro, showing that both resting and activated mast cells induce osteoblastic remodeling of smooth mast cells and induce macrocalcification. However, when the macrocalcification is established, it's capable to attenuate the mast cell activation. In summary, integrative analysis of clinical and molecular data were able to identify from study 1 a new link between macrocalcification and amyloid structures. Study 2 and 3 revealed osteomodulin as a new marker for vascular macrocalcification, and it was able to link high osteomodulin levels with fewer cardiovascular events. And study 4 revealed an association between mast cell phenotype and macrocalcification. Here, I would like to thank my promoters, both in Maastricht University and in Karolinska, all my colleagues and the collaborators that participated in those studies. Thank you for your attention. I will now give the floor back to the prorector.
Thank you very much, Mr. Skenteris, for your clear presentation. The opposition will be opened by the chair of the Thesis Assessment Committee, Professor Casper Schalkwijk, and he is Professor of Experimental Internal Medicine. Professor Schalkwijk.
Thank you, Mr. Chairman. Dear candidate, first of all, of course, I would like to congratulate you with this excellent thesis. And for me, it was really a pleasure to read it. And I'm really impressed by the relevance and the quality of the work based on several state-of-the-art techniques. So once again, my congratulations. So we have focused on different forms of vascular calcification, the role of smooth muscle cells, the underlying mechanisms. And I've learned actually from your thesis that there are different forms of calcification with beneficial effects, with harmful effects, and but so also with beneficial effects. So I'm a little bit confused about your first sentence, actually, in the abstract, and you referred actually to publications demonstrating that calcification, the detection of calcification, is actually a risk predictor for all-cause mortality and also for cardiovascular disease. But how to explain that? Because some of the aspects of calcification are actually beneficial effects. So how to explain this in relation to, let's say, the literature?
Hi, esteemed opponent. Thank you very much for your question. I would say that the majority of publications in the literature summarize the calcification in coronary arteries disease and also in aortic valve disease. And in that, those two diseases, calcification actually is detrimental and has been linked actually the high calcium score in the coronary arteries with more increased disease mortality. And at the same time, in aortic valve leaflets, the higher calcification score actually inside the valves, it causes valve leaflet impairment. So in those two situations, calcification is like rather detrimental than beneficial. However, studies in our lab have shown that calcification, and particularly macrocalcification in carotid atherosclerotic plaques, it's rather a protective and stabilizing mechanism. Since it inhibits the rupture of the carotid plaques. And also I would like to pinpoint that here in the study we have, in the thesis, we have studied macrocalcification compared to microcalcification. And microcalcification actually has been linked with inflammation, and it has been found in the atherosclerotic plaques in the fibrous cap. And actually it causes destabilization of the fibrous cap and the rupture of this plaque. Mm-hmm. So we have to, I think we have to classify these two forms of microcalcification and then according to what kind of disease we discuss. We have this classification of macro classification between carotid atherosclerosis or coronary artery disease and valve disease.
Okay, that's clear. Thank you. So you found actually also in the first study an unexpected link, I think, with beta amyloid. So more or less the same underlying mechanism, activation of platelets, release of compounds, the same for calcification as for beta amyloid.
Mm-hmm.
Based on that, just wondering what is known about calcification in Alzheimer's disease, or what is known about, let's say, platelet activation in Alzheimer's disease? Have you found something about that, or what's your idea about that?
Yeah, thank you very much for the question. I don't think that there is so much literature on calcification in Alzheimer's disease. However, I think that platelet activation is participate also in amyloid deposition in the Alzheimer's disease inside to form the amyloid plaques. Since these beta amyloids, particularly in the brain, in the Alzheimer's disease, is amyloid 42 molecule, which is released either from microglia. It's actually in the surface of the cells or in the surface of platelets, and cleave of this alpha B42 peptide is participated to Alzheimer's disease. In our study, also we couldn't find any link between actually epidemiological link between Alzheimer's disease and aortic stenosis. Okay. There's nothing in the literature. However, there is this new notion in the field of aging heart, or this that links the Alzheimer's disease with the cardiovascular disease actually. Mm-hmm. So there is this new notion in the field, in the cardiovascular field, and also there is this TTR aggregation that causes the amyloidosis in the heart. Mm-hmm. Mm-hmm. But here we were able to link the calcification, particular calcification, TTR aggregations with calcification.
Okay, so your idea actually is that the release of amyloid precursor proteins, APP, is directly involved actually in the process of calcification. Is that true, or?
We don't know yet, because what we have studied is that it's publicly available data and it's end-stage disease. So we don't know if the APP molecule participates from the beginning in the mineralization process in the aortic valves, or it's like an end-stage process when you have the impairment of valve leaflets, and this impairment of valve leaflets actually causes the activation of.
But if that is true, have you any idea about the underlying mechanism of APP in relation to the induction of calcification?
There is studies showing that platelet activation actually from a lab in Canada, showing that platelet activation causes mineralization in VIX, in vitro and in vivo. So apparently what platelets secrete, or the platelet activation and degranulation, can cause the mineralization process in valvular and interstitial cells.
Mm-hmm. And so in actually your last chapter, you studied mast cells, and more or less also the secretion of granulates from mast cells are involved directly in the calcification process. Do you think that also here APP is of importance? What are actually the components in the released granulates which are of importance in relation to calcification, so release from mast cells?
APP in the circulation has been found only in platelets, not in other immune cell type or immune cell. So it's only in platelets. However, regarding your question, the mast cells actually have packed in those granules neutral proteases mainly, and metalloproteases, tryptase, chymase, and all these proteases. However, the activation of mast cells can release those or secrete these proteases in the extracellular matrix, and can cause the degradation of extracellular matrix. Okay. In addition, the activation of mast cells can release growth factors such as TGF-beta or FGF, and several cytokines like pro-inflammatory cytokines, interleukin-1 beta, IL-6. So we're not sure what it's releasing from activated mast cells that cause the mineralization process in the smooth mast cells.
Okay. Thank you so much. I'm really satisfied with the answers, and I give the word back to the pro-rector. Thank you so much.
Thank you very much, Professor Schalkweg. The opposition will be continued by Professor Alan Schirschö. He is a professor of medical surgery at the Cardiovascular Research Institute at Örebro University in Sweden. Please unmute your microphone.
Now, thank you, Mr. Chairman, dear candidate. As you probably know, there is quite good relationship between lipids and the development of atherosclerosis. What's your opinion about lipids and calcification?
I think there is a direct link between lipids and calcification, because lipids are and I suppose the oxidized or modified LDL particles actually induce smooth mast cell reprogramming towards osteoblastic differentiation. This is already known in the field, and so the taking up of those oxidized lipids by smooth mast cells induce the osteoblastic reprogramming of smooth mast cells. And at the same time, I suppose that lipids trapped in proteoglycans, which are secreted from smooth mast cells, and this also enhance the mineralization process of the matrix mineralization process. So I think there is a direct effect between oxidized lipids or lipids in general. However, there are studies showing that HDL and recombinant HDL particles can attenuate the mineralization process.
Okay, because you can find vessels that are highly calcified and no lipids, I suppose.
Absolutely, but this is quite interesting in medial calcification when you don't have the inflammation or lipid engagement in the mineralization process. However, those vessels, the medial part of the vessel, is mineralized actually. However, this is a different mechanism. The smooth mast cells, due to the excess of phosphate, become apoptotic, and this exacerbates the mineralization process or release the calcified vesicles, and which lead to the mineralization process. And this is particularly obvious in patients with chronic kidney disease and diabetes.
Okay, because I think you find a negative correlation to osteomodulin and lipids in the vessel, didn't you? Or?
Yes.
So that means less lipids, more calcification, I suppose.
Yes, but I think the table that you are mentioning is from patients with chronic kidney disease. So it's exactly the procedure that I explained. In those chronic kidney disease patients, we see middle kind of calcification.
Okay.
And there we have measured the plasma osteomodulin levels, and we were unable to correlate with lipids level.
Okay. Then I just have a question about your experimental setup, especially with smooth muscle cells in culture. If I understand it right, you buy your smooth muscle cells from some company.
Yes.
And then if we say that you have one control group and one stimulated group, is the number of experiments, is that different passages? Or is it the same passage?
Highly esteemed upon, thank you for the question. We buy the cells, the smooth mast cells, however, from a company, however, it's primary, aortic smooth mast cells, the one that we have used. So it's from the patients that have transplantations, and it's like healthy smooth mast cells. And we treat smooth mast cells with either high phosphate or beta-glyphosate, and we have untreated control. And we passage from, we have triplicates, biological triplicates, but those biological triplicates in different passages. Like we don't play in the same passage the whole experimental setup.
Okay. So the number of donors here is that 3?
The number of donors is 1, but we have cells in different passages.
But do you think, so the results is from one person really? So it's built on how that person's genes are expressed, I suppose.
That's true for the cells used from the company. However, the same experimental setup has done in Leon Serger's lab in Maastricht. And there we used cells, thoracic aortic smooth muscle cells from different donors. So we have used three different donors there. And we actually were able to replicate the same results.
Okay, because yeah, my experience is that you can get very different results from different donors.
Absolutely, you have absolutely true.
What's more? Yeah, you find, if I understood it right, in this table, you have looked at the message level and looked between asymptomatic and symptomatic. And on message level, you find high levels in asymptomatic of this osteomoduli.
Mm-hmm.
But on protein level, you could not see that. Is that correct, or have I misunderstood
it? On protein level, we didn't check. This is the tissue mRNA levels of osteomodulin, and it was the levels were upregulated in asymptomatic patients compared to symptomatic, and then we linked those with future cardiovascular events or cardiovascular death. But we didn't check the protein levels of osteomodulin in symptomatic versus symptomatic.
Okay. Okay, thank you. And I will leave the word back to Pro-Rector.
Thank you, Professor Sircher. The opposition will be continued by Dr. Nylin Lee. He is Associate Professor at the Division of Cardiovascular Medicine of the Department of Medicine of Karolinska Institute in Sweden. Dr. Lee.
So, thank you, Mr. Chairman. So, dear candidates, first I would like to congratulate you for the great thesis. I enjoyed reading it, and since I'm already in your half-time control, I really feel that you have done a lot during the last 2 years. You do a great job and write a nice thesis. I really enjoyed reading it. And I think in your thesis, you mentioned many places that the mast cells are very important for the vascular calcification, and you also use your studies and show that the paper 4 that mast cell activation really close relate to the calcification. Could you a little bit further delineate how mast cells could contribute to the vascular calcification? How mast cells contribute to these processes?
Highly esteemed opponent, thank you very much for your question. Indeed, the mast cells, it's a small proportion of the cells that are found in the plaques. However, it's like a big effectors. So, and this is due to the effect that release those granules, and when they become activated, they release many cytokines, pro-inflammatory cytokines or anti-inflammatory cytokines, and other like lipid mediators and other molecules like growth factors. And we assume, and actually there are studies showing that those extracellular matrix is degraded by those proteases that the mast cells release through the degranulation process. And those proteases are like cathepsin S, which has been linked with degradation of extracellular matrix, MMP2, which is breaks down the elastin, and also MMP2 has been linked with the mineralization process. And we assume, even though we haven't found a direct link or a direct molecule that associates directly with the smooth muscle osteoblastic reprogramming and matrix calcification, we assume that the combination of several molecules actually participate in this osteoblastic remodeling, osteoblastic transition of smooth muscles and remodeling of extracellular matrix towards mineralization.
The muscle also secretes serotonin as well, right?
That's true. They secrete also serotonin.
Mm-hmm.
And also serotonin has been linked with the calcification process. There are studies showing this in the valve leaflets, actually, and the aortic stenotic patients.
Do you see the major increase of the mast cells present in the calcified tissue? Yeah.
We saw that resting mast cells actually are upregulated in or are enriched in highly calcified plaques.
Okay.
The activation of mast cells take place in low calcified plaques, actually.
And also in the introduction of your thesis, you stress that platelet drive, the TGF-beta, is very important for the transforming the smooth muscle or these calcification cells. Why do you think or the platelet drive transforming growth factor beta is so important for these processes?
Thank you for your question. TGF-beta is like a BMP2 family molecule, and it has been shown that TGF-beta actually induces MAD3 upregulation, which several studies also from our lab and others have shown that upregulates the RANX2 and SOX9 transcription factors, which actually master regulators for osteoblastic transition of smooth muscle cells. And I think that's why also in our studies we have used TGF-beta in order to induce in some experiments osteoblastic remodeling of reprogramming of smooth muscle cells. And
yeah, please go ahead.
And since it's belonging to the BMP family, it's also BMP2 is actually capable to induce this osteoblastic reprogramming of smooth muscle cells. So it's two different molecules under the same family. Okay,
because the TGF-beta were produced by multiple cells, but do you have solid evidence that shows that from the literature or from your own data that it is platelet-derived TGF-beta is important for the vascular classification?
Indeed, TGF-beta is released by several pro-inflammatory molecules like mast cells, platelets also release the TGF-beta, and here it's only, we speculate that this might be the link between, if you are referring for the fourth study, mast cells, we speculate that TGF-beta might be a molecule, but this is not like in solid experimental results.
Okay, so I really liked that you did a multi-omics integration analysis that use the public data, and I do see you have a quite intensive discussion how to use this data, and could you, because I also see you have different method to use this to make this heterogenic data comparable and make the comparison analysis meaningful. But still, I see that you comment when you have this, you see the OS expression of this complementary and co-creation system is you only see the positive data from 4 out of 20. It's still quite low number too in my eyes. Is there any ways you could think for the future to improve this, to make this comparison or the analysis more useful and bring out more pure data?
Yeah, that's true. Indeed, in our analysis of study one, we used quite diverse, I would say, data sets, since it's publicly available data, and Here we used only the molecules that are described in the papers, or we asked for the authors to provide the raw data. However, we didn't make any classification between upregulated and downregulated molecules. Therefore, we describe as dysregulated molecules. And in our pathway analysis, we found complement and coagulation as dysregulated pathway, and we don't know if it's upregulated or downregulated in the calcific aortic valve disease patients. So I would rather suggest for future studies to have like multi-omic analysis from the same patients and integrate the data from the same patients, let's say the proteomics, transcriptomics, lipidomics, and metabolomics data from the same patients, and link those data with clinical data sets and medical records for the patients, and then would be more able actually to link the signature, the molecular signature of the same patients with the clinical symptomatology of the patient.
Okay, thank you very much. Thank you. So,
thank you, Dr. Lin. The opposition will be continued by Professor Judith Sluimer. She is Professor of Cardiovascular Pathophysiology at Maastricht University. Professor Sluimer.
Thank you, Prorector. Dear candidates, also I'd like to congratulate you with your thesis. Happy to see you, not live like you were in a lab, but still nice to see you. And I'd also like to congratulate your team on two sides, or from two institutes, with this nice accomplishments, despite being really troubled, of course, by the pandemic. I would like to talk to you a little bit about the regulatory link between osteomodulin and calcification. So in your figure 17, you describe that OMD is lower in symptomatic plaques, but higher in calcified plaques, and that the higher OMD is associated with less events. And I was kind of puzzled by these different associations, and I was wondering how you perceive or do you perceive a relationship of OMD beyond just being a biomarker, or do you think there's a regulatory connection there?
Hi, listening opponent, thank you very much for your question. Indeed, we were able to link osteomodulin mRNA expression, you said in figure 17. Yeah, with calcification process, actually it's upregulate. It's a positive correlation between calcification and osteomodulin, and we suppose actually we have found also from data from chronic kidney disease, we found a direct link between osteomodulin plasma levels and calcification score in a valve of chronic kidney disease patients, and then we went back in patients with calcific aortic valve disease, and we detected the osteomodulin there. So there is a direct link between osteomodulin and calcification, and since it's a proteoglycan, extracellular matrix proteoglycan, and has been also described in the literature that it participates in the mineralization process both in bones and in teeth, we suppose that it has a active process in the mineralization, and here we found that it might be like an early marker for osteoblast differentiation rather than as a late biomarker for calcification.
But dear candidate, what do you then think is a chicken or the egg? Do you think that the high OMD and high calcification is because there is more calcification, or do you think that OMD actually is causing the calcification? And that's not clear to me.
Actually, what we have shown from the in vitro datasets, and also we have correlated with data from the plaques, so we have seen that the osteomodulin is quite upregulated through inflammation process. And since inflammation, both IL-4 interferon gamma and IL-6 also is induce osteomodulin mRNA expression. And since inflammation process is quite early during atherosclerosis progression, we suppose that these pro-inflammatory stimuli actually induce the smooth muscle cells to release this proteoglycan. And also the proteoglycan participates in the extracellular matrix remodeling. So we suppose that it has an active role in the extracellular matrix modeling, and quite early on during the disease progression.
Okay, well, that brings me to my next question, because I was already thinking if there might be any confounding factors in this association that you're proposing, and I assume that your survival plot is corrected with age, sex, and lipid levels. But did you also correct then for inflammatory levels in this survival plot?
No,
Would you consider correcting for this or for any other aspects?
Yeah, that would be a nice addition, actually.
And just for my curiosity, did you also make a survival plot with the high and the low calcifications? And do you see there the same pattern then?
No, actually, we couldn't see. At least in our data cohort from BIKE, we couldn't see any relation between when we classify high and low levels of osteomodulin in the plasma, we couldn't see any connection between calcification, actually.
Okay, not in this cohort then. And as you're already alluding to, you said that the osteogenic cells may be expressing more osteomodulin. So then, do you think this causes the reduction of osteomodulin in symptomatic plaques? Do you think is it related to the content of those osteogenic cells?
We assume though, since in symptomatic plaques they have less osteoblastic or osteochondrogenic smooth muscle cells, we assume that since it's less and it's highly inflamed plaques usually lead to symptomatology in the patients. So we assume that since there are not so many osteochondrogenic smooth muscle cells, they have less secretion of proteoglycan. So that's why we see less.
But do you think it's less per cell, or is it because there's just less cells?
Both, maybe.
Okay, maybe in future you can.
We can further investigate actually this, and it will be very nice also to investigate further this relation between plasma osteomodulin and calcification in large cohorts actually with more patients, and see if there can be used like as a clinical biomarker to detect early on the calcification process in patients with cardiovascular comorbidities.
Yeah, all right. Thank you very much. I'm happy with the answers. I give the word back to the prorector.
Thank you very much, Professor Sluimer. The opposition will be continued by Dr. Rory Koenen. He is Associate Professor of Biochemistry at Maastricht University. Dr. Koenen.
Yes, dear candidate, thank you, Prorector. Dear candidate, I also enjoyed reading your thesis a lot, and I want to congratulate you and your team for this achievement. Without further ado, I would move on to the questions. A couple of topics have been covered that I want to cover with you, but there's still some room for discussion. So you have in your chapters, mainly in study 2 and 3, you've looked at osteomodulin messenger RNA and osteomodulin protein, and then of course a different form. So just some basic questions. How well does messenger RNA correlate with osteomodulin protein? Is that direct in your plugs?
We haven't checked actually because we didn't measure with proteomics data the osteomodulin mRNA expression. So to have a direct and see the correlation, if there's linear correlation between the transcriptomics and proteomics data for this particular molecule. However, as you appreciated from the figures, we could detect both intracellularly and extracellularly the osteomodulin staining in low and high calcified plaques. So the protein is abundant there in the tissue.
So messenger RNA means proteins. Okay, and then the protein is sort of bound to the extracellular matrix, and that's what I understood. It's sort of an intimate connection. Mm-hmm. And then you also have soluble protein. That's what you measure also, I think, both chapters 2 and 3, or studies 2 and 3. Where does it come from? The soluble.
The soluble, we assume that it comes either from the tissue or from the bones, because also bones synthesize osteoblasts there in the bones synthesize and release osteomodulin. So, and since we have this paradox actually in the calcification that osteoporosis leads to higher vascular calcification, maybe it's released from the bones, and we have this upregulation in the circulation of osteomodulin protein coming from there. we're not sure.
I have another thought I want to share with you. Because it's so strongly associated with the extracellular matrix, maybe it is released when extracellular matrix is broken down. Would that be an option? Because that would, in my opinion, have also a related link to stability then.
Actually, the osteomodulin has been found that it's linked with collagen, and also there is studies showing that elastin degradation actually induces smooth muscle osteoblastic remodeling. And maybe this elastin degradation through other processes or metalloproteases that degrade the elastin composition induce this osteoblastic remodeling of smooth cells, and then we have induce the upregulation of both mRNA and protein inside the tissue. However, in the circulation, we're not sure how this protein actually released in the circulation.
Thanks. That is clear to me. Thank you very much. So, and also with Professor Sluimer's question, I want to take it one step ahead. We've talked a lot about stability of plaques and about symptomaticness of plaques, let's call it like that. But aren't you focused too much on plaque rupture, actually? Because you mentioned in your proposition 7, I'm not going to ask you to read it because it's very long, but basically the scientists that you cite in that proposition, they are saying that plaque erosion is actually becoming more and more important. What could you say about the relationship between calcification and plaque erosion?
Plaque erosion you have when the endothelial cells are denatured. So you have in the circulation you have the smooth muscle directly that connected with the circulation actually, and the platelet is attached there. So I assume the platelets that attach directly on the smooth muscles can induce a smooth muscle differentiation there, and then you can have this like maybe amyloid-like structures that we have found from study 1. And
could it also be that, for example, plaque erosion, because if you have a calcified plaque, that it might erode more quickly than a non-calcified plaque, and that maybe also one little link to the paradox, I don't want to call it paradox, but to calcification actually is a risk factor for events.
It has to do with the calcification, actually, the location and where the calcification is localized. And what we have seen from our studies is that calcification is localized in media and intima side of the plaques compared to the fibrous cap. So in the fibrous cap, you have usually the microcalcification, that because this region is quite inflamed, highly inflamed, and you have the microcalcification. So maybe the microcalcification there is linked with the plaque erosion rather than the macrocalcification. Okay, so I have one question. Maybe we can do it shortly. Let's see. I was just wondering, you're in your study 3, I think it's study 3. You cultured smooth muscle cells in an osteogenic medium, and then you get osteomodulin expression. And then in your RNA sequencing data set, that's in study 2, you see that it's actually not the smooth muscle cells expressing osteomodulin, but fibromyocytes. Yeah. I was just wondering, when I all read this, what is actually a smooth muscle cell? Is a smooth muscle cell you have in your dish the same one as you have in your plaques? Because the smooth muscle cell, as we know, has a lot of different phases. How do you put this all together? Mm-hmm. Thanks very much for the question. I would say that in plaques, we see ACTA2 positive cells, but really smooth muscle cells are embedded in the media layer rather than in the plaques. And the plaques are quite heterogeneous composition. So it's already modulated smooth muscle cells rather than smooth muscle cells. That's why we also see from the single-cell RNA data that it's already modulated smooth muscle cells, and the researchers there named those are fibroblasts-like or fibrochondrocyte-like cells. So of course, those two types of cells, they express also ACTA2, that which is like an origin of those those modulated smooth muscle cells come from smooth muscle cells. But I wouldn't say that in the plaques you have a homogeneous population of smooth muscle cells in the plaque, inside the plaque. It's already modulated smooth muscle cells.
Yeah. Okay. Yeah, I think we have to stop the discussion, but I'm very pleased with your answers, and I would like to give the word back to the prorector.
Thank you, Dr. Koenen. The next opponent is Dr. Ilse Bot. She is Associate Professor at the Division of Biotherapeutics at Leiden University in the Netherlands. Dr. Bot.
Thank you, Prorector. Dear candidate, first of all, I would also like to congratulate you with your thesis. I read it with great pleasure, and also would like to congratulate your team of supervisors. It was a nice piece of work. And maybe not surprising, I would like to discuss with you study 4. But not before I want to ask you to read one of your propositions, which is proposition number 4.
I don't have it in front of me right now.
Okay, I can read it for you. Your proposition number 4 is the activation state of vascular smooth mast cells reflects the calcification degree of an atherosclerotic plaque. That's what you proposed. So I was wondering, because you in your study you did like an association between your calcification and your smooth muscle cells calcifications. Why did you not formulate your proposition as the calcification degree of the atherosclerotic plaque reflects the activation state of your mast cells?
Would be other way around as well, but here we don't know from the data set, since we studied the end-stage calcific valve disease, atherosclerotic patients. We don't know if the activation status is happening earlier or later on. However, from the in vitro data showing that both resting and activated muscles actually induce smooth muscle reprogramming and matrix calcification. And when this matrix calcification is established, we have the macrocalcification is established in the extracellular matrix. This is capable then to attenuate the muscle activation. So we don't know if the way that is happening inside the plaque tissue.
So it could go both ways, like your mast cell activation can reduce your calcifications, but your calcifications can also inhibit your muscle activation.
The muscles they cannot reduce the calcification process, because both resting and activated muscles induce matrix calcification. However, the calcification process or the calcified matrix can attenuate the activation status of mast cell.
And how do you think that works then? What's in the, for instance, you did your in vitro experiments where you stimulated your mast cells with supernated from advanced non-calcified plaques versus your calcified plaques. What's in the non-calcified plaques that can activate your mast cells, which is not present in your calcified plaques?
The non-calcified plaques are abundant with lipids, and since the calcification degree there is quite low, they have enlarged the lipids necrotic core and maybe pro-inflammatory stimuli that enhance the activation status of mast cells. So
you think it's like the pro-inflammatory environment, but you measured mast cell degranulation by your staining, for example, and that's an active process which occurs via specific and dedicated receptors, for instance, IgE, Fc epsilon receptors. What do you think is, can you speculate on what's maybe the most prominent mast cell activation mechanism there?
I would say pro-inflammatory cytokines, because mast cells also have TLR4, and the low calcified plaques are enriched with lipids necrotic core and mast cell apoptosis, macrophage apoptosis, and pro-inflammatory molecular apoptosis, which release cytokines that activate TLR4. And I would say that the activation happening through TLR4 or those molecules rather than Ig and Fc receptor 1.
I would agree on that. But if you then go to your in vitro experiments where you take the supernatant of your smooth muscle cells that you actually grown in calcification medium or not, also there you see increased mast cell activation when you expose your mast cells to supernatant of non-calcified smooth muscle cells. Do you also see increased inflammatory mediators there in that supernatant as compared to your calcified smooth muscle cells?
Yeah, I don't really know, because even though those smooth muscle cells are, we have challenged them with either control or high phosphate. Actually, we have seen the same results as in vivo, that supernatants that come from from calcified smooth muscle cells attenuate muscle activation compared to control. But we don't know what is coming in the control supernatants that have higher higher muscle activation actually.
It may be good to look at the inflammatory markers there.
That's a good addition.
Yeah. So if you want to gain more mechanistic insight, you could propose to do like an in vivo experiment where you look at the relation between mast cells and calcifications. Can you think of such an in vivo experiment where you can definitely pinpoint which mechanism is going on there?
I would say to use muscle stabilizers in mouse models with warfarin, which and a high-fat diet, which induce actually vascular calcification in the mouse model, and at the same time have muscle stabilizers, or the other way around, to see what's induced, if the mast cells activation or the stabilization of mast cells attenuate the calcification process in the mouse models, or it's the calcification process that attenuates the muscle activation. So it's I would rather very sophisticated in vitro experiments that would illuminate further this interplay between muscle and activation and macrocalcification.
And do you see many calcifications in like mouse atherosclerotic plaques?
We don't see at all. That's why we should use warfarin to induce extracellular matrix mineralization in the arteries.
And if you think of aged mice, because in humans also decalcifications progress upon age, is an aging mouse model maybe a better way or a less artificial way to induce calcification?
I would say that it's a rather different mechanism, because in aged mice we have like medial calcification, which is not triggered by inflammation, and you have this arterial stiffening there. So the medial calcification induced the aging, the arterial stiffening, and in aged mice. However, for atherosclerotic context, we need to have plaques, which it's induced by high-fat diet, and atherosclerosis, which is induced by warfarin in the mice. So I would rather go for muscle stabilizers and ApoE background mice fed with high-fat diet and maybe some warfarin and stabilizers, muscle stabilizers, rather than in aged mice, because the mechanism, the calcification mechanism, is completely different there.
Okay, so then maybe one final question: how can you translate the findings that you have in this experiment into like a therapeutic application for patients? Is there a therapeutic opportunity?
For a study for you mean?
Yes.
I would rather, because here we used imaging and transcriptomic analysis to use.
Yeah, Mr. Skenteris, as you have seen, and you must have noticed it, that the time for defending your thesis has passed. The committee will now withdraw to discuss the quality of your thesis and the quality of your defense. And please await our return with the results of our deliberation. Mr. Skenteris, the degree committee here present online has assessed the quality of your thesis and the quality of your defense. And in view of its positive verdict, and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Biese is authorized to confer upon you this academic distinction in accordance with Dutch university custom and law, and invite your supervisor to take the floor. Professor Biese.
So first of all, I have a statement to claim. So, do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
I do promise.
By the authority vested in us by law and in conformity with the decision of the committee here present online, I hereby confer upon you, Nikolaos Taxiarchis Skenteris, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, you will soon receive the degree certificate signed by the rector, the secretary, and the supervisor affixed with the official seal of the university, shown now by the beadle.
Thanks a lot.
Then, Dr. Skenteris, may I be the first to congratulate you with the newly acquired title. It has been a pleasure, although a bit plagued by the COVID pandemic, to have you in our lab for 6 months. And I got to know you as a very ambitious, talented, and also a bit of restrained scientist that has developed now, as you have shown now, in the past hour, into a very mature scientist. So I wish you all success in your further career and hope it will be in science. But now I give for the laudatio the word to Hilde and Lubika, your main supervisors.
Thank you, Professor Biese, and thank you for giving us the word. I'm very happy to congratulate our PhD candidate and new PhD, Nikos, on this excellent thesis defense. And furthermore, to congratulate him on the whole challenging PhD road that he has taken when he stepped onto Karolinska. And actually, to thank him for choosing us as his supervisors and co-supervisors, because it has really been a pleasure to work with you, Nikos, over the past few years. Like I said already, and like we have discussed many times, it is a challenging road, but you really have shown decisiveness, persistence, and creativity that is necessary to come to this point in time to actually become a PhD. This is a big lifetime achievement, and I hope you are going to enjoy it. At the end, I also really want to especially thank the whole IntriCare program who has brought us together here in a joint effort between Maastricht University and your supervisors at the Maastricht University and Karolinska. This has resulted in an excellent collaboration that we can only hope it will continue in the future and produce more collaborative studies and PhD students and other students as well. So thanks a lot to everybody, and especially to Nikos for highlighting this day for all of us. Professor Arnardottir, I hope you can take over now.
Yes, dear Nikos, let me start by congratulating you on your absolute excellent defense today. And this is the first milestone towards your double degree with Maastricht University and Karolinska. Now there's one in the back, Dr. Nikos, and only one to go until you reach the end of this journey. And like Lubika said, what a journey it has been. As with life, it hasn't been without its challenges. But you know what they say, challenges build character. But I would like to challenge that, and I would say it rather reveals character. And yours has really shown throughout this whole journey, not just your determination, your perseverance, your passion, and your ambition towards reaching your goal, which is now just at your fingertip, but also your personality, your friendliness, your warmth, and your curious mind, which has allowed you to build up a fantastic network of both friends and collaborators that will continue to grow with you into your future endeavors, wherever that may lead you. And you really took the best out of every scenario, and you kept moving upward and onward. And here we are. And on these type of big moments, one finds one's reflecting back, and I'm no exception. I still remember when I first met you in CMM in October 2018. I just returned back from maternity leave, and you had joined the group while I was away. And even though we didn't know each other at the time, you seemed very happy to see me, which I also now realized later that it was just your normal, friendly and warm personality, but you immediately said, "Hi, I've been waiting for you." Or that is at least how I remember it. But there may have been words exchanged first, but I must admit, I was a little surprised, but of course honored that the new guy that I hadn't met before was waiting for me. But of course, I was part of your supervisor team at the time, and you had been working on a project that aligned closely with mine, and you had really made some excellent progress, and I was really impressed when I came back. But at the time, you felt like you needed more support and guidance. This is why you've been waiting for me. And I do remember we had an immediate connection, both on a personal and a scientific level. And I realized very quickly that we would get along well. And I was really happy to be able to provide you with the support that you needed at the time. And although you may not have found a home within our group, I'm so happy that I got the honor to continue with you on this journey together with Lubica, where you not only found your scientific home, but you really flourished and developed into the confident PhD that we witnessed here today. So again, I would like to wish you my biggest congratulations on your today's fantastic achievement. And I look forward to keep marching with you through this grand finale of your PhD journey and to the final celebration on Friday. Thank you.
Thanks a lot.
Esteemed Dr. Skenteris, dear Nikos, it's my great pleasure to congratulate you with your doctorate and the honor that you have acquired today. And I do that also on behalf of the Board of Deans of Maastricht University. And now I would like to share some impressions of the committee, looking back at your thesis and your defense. And we have seen and we have enjoyed an excellent and a clear presentation. And the thesis was very good. It was a relevant subject, but also a challenging subject. And we have seen also that you have shown that you master quite a variety of techniques, which was mentioned as almost complete. And we are more than satisfied with your defense. High quality, outspoken, passionate, and creative. And you have shown that you are familiar with the subject, you have a good knowledge, and you showed a good and a high academic level of defense and discussion. So I would like to congratulate you with that, but also your 4 supervisors. Professor Erik Biese, Professor Chris Reutlingsberger, Assistant Professor Arno Dottier, and Associate Professor Lubica Matic. And I congratulate you all 4 as a team with the excellent results that we have seen in the form of a thesis, good publications, and a very good defense. And of course, I include in my congratulations your family and your friends, and all those who have contributed to making this thesis and allowing to present this this presentation, but also a good defense that we saw today. So congratulate I would like to congratulate all these people with your success, and I wish you a very successful professional career and, of course, a happy personal life. And before I close this academic session, I would like to thank all the members of both this degree committee and the assessment committee, and in particular the external members. And I would like to thank all the opponents and the supervisors for their interesting academic discussions and their input to making able this defense today. And Maastricht University appreciates your contributions. And I also want to like to thank the supporters of this academic ceremony, the ICT supporter, Mr. Luc Peters, and the members of the PhD office and the Beadle. And now, hereby, I close this academic ceremony.